Welcome back, friends. In this video tutorial we'll be talking about inflammation, which is a classic example of immune system in activity, right? And this is one of the most important thing that you must understand about the immunology. This is the very first and most important thing I'm emphasizing. Okay, now let's talk about inflammation. Inflammation is the very first response. It's actually the first response of your immune system cell against a kind of foreign invader or a foreign pathogen, right? Now there are typical five signs of inflammation that we can see here. These are the different five signs. This is the cartonic representation that is telling us, and we all witness inflammation somewhere many times in our lifespan. Any kind of infection or injury, whatever injury that we get, these five signs like heat, redness, swelling, pain, and loss of function, so all of them are associated with each other, and all of these facts that you are getting after any kind of injury, you can see here the color is becoming red and it is swelling, then loss of function, and this particular part scab is formed and all these things. So all of this process, you may wonder many times why it is happening, why all these things are happening. And all of these things, in most of the cases we see that not only not only simple inflammation, not simple, but you can say swelling, we see all this thing all together. What it means then all of these processes are linked with each other. That's the major important point I want to focus on. All of these things are linked with each other, and they are linked via the process called inflammation. Now inflammation is a process, it indicates, it tell your immune system, which is just resting in relaxed state, it is telling you that something is going wrong, you need to come and watch for this, right? Now as it is telling all of these cells, that's why this inflammation is very important. This is very important, and very first round of immune response. Now, usually the inflammatory process that we usually deal with are of two different type. One is the acute phase of response, another one is the chronic phase of response. In the acute phase of response, we are talking about the quick response, because in this case we need to respond to the infection or injury very fast. But if we look at some kind of infections which are chronic type of infections, where the pathogen plays and stays there for long period of time, we'll be seeing this chronic inflammatory response. Now let's first talk about the acute phase of response. We can also divide the acute phase of response in two different parts. One is the localized response, which is the very beginning of the response, at the localized place, at the tissue where the injury occurs. And another response is also there, which is called the systemic response. But I must tell you that the systemic response come later of this localized response, because localized response obviously come first, and after the localized response is established totally, then it can be systemic, it can spread from one place to another place of the injury, and then it can only become systemic. But rest of the place, the first important response is localized response. In the localized response, we can witness those five different symptoms: swelling, redness, heat, pain, and loss of function. And in systemic response, except for all these things, it responds some more level of expression, and the expression is induction of fever, an increase in WBC count, and acute phase proteins. And also in chronic inflammation, what we can get different type of expressions, like different you can say again induction of fever, and also increased number of WBC and natural killer cells and macrophages in our bloodstream, so that we can understand that something wrong is going on all the time. That's why our immune system is triggered. And usually this kind of chronic inflammation is seen in case of some dangerous disease like tuberculosis, mycobacterium, caused by mycobacterium uh, tuberculosis bacteria. And obviously in case of cancers and in case of autoimmune diseases, we can find this chronic inflammation. Now let's move on to the first important or concept about the plasma inflammation mediators. We need to talk about these mediators first, because if I talk about mediators, the mediators are some important components that we can see, which are provided, which are produced after any kind of endothelial line damage. So endothelial cells are there in in our, what you can say, blood vessel. Now this endothelial cell damage, whenever this endothelial cell damage occurs, now this endothelial cell damage is required, because remember, let's say here, let's say this is a tissue, so let's say this is a bloodstream. Now the bloodstream is moving on, and somewhere here is our tissue of interest. Now somewhere here we are having the infection, we are having pathogen sitting on there, they are having fun, so we need to go against this infection. So for for call upon all the other immune cells, we need to take them out from bloodstream, because rest of the immune cells, if I draw them with this blue color, rest of the immune cells are present in this blood vessel. So we need to make a hole on the blood vessel, and we need to drag these immune cells like macrophages, natural cells, T cells, B cells, to come at this place and recognize this particular type of infection or infection causing agent, and then go against them, right? So for that reason, what we require, we need vasodilatation, right? So this is required. If in the previous case, if this is the diameter of our uh, vein, now it should be broadened. And obviously not only broadened, but also they must create the holes in between the epithelium cell lining, right? So any kind of damage during this process must be necessary, and this kind of damage are established by several kind of chemical mediators or chemical factors, like histamine is one of them. So histamine is one of those chemical factors that we are going to see. Okay. Okay. Now let's say here in this case, after the endothelial damage, what it produces, it actually produces major three different components, and one of the component is plasmine, second one is fibrin, and third one is bradykinin. So these are the three important components that are produced after the endothelial cell damage or endothelial cell lining damage. Now among all of them, they are having specific functions. Like bradykinin has the function of vascular permeability, which I have talked about, the making holes on in this vesicular endothelium. So this increases the vascular permeability, and obviously it causes some pain and smooth muscle contraction. And there are other components like fibrin or fibrin peptides, and these peptides help in fibrin degradation. Now when the fibrin degradation occurs, this fibrin degradation help in the clotting of blood. So this is the important part, it helps in clotting of blood. And the third thing is the plasmin, and the plasmin helps in complement activation, because complement activation is required to kill cells or to kill pathogens, not only cell, but these cells of pathogens. Actually I must write pathogen here. Okay, so to kill pathogens. So these are the three different mediators that are produced after the inflammation, first round of inflammation. Now let us first talk about the local inflammatory response. Now in the local inflammatory response, what is going on is that we are having the bloodstream, and this bloodstream is having all these components like macrophages, neutrophils, and all these other important components or immune system cells which are fighting against this immune system, right? And obviously these are associated tissues just surrounded by these blood vessels. Now there is something going on in this tissue, right? For example, let's say this is the first type of response where bacteria invades in, and they are causing some problems. Now as they are invading in, there are other components which are called macrophages, which are called mast cells, which are sitting there, which are simply sitting on the tissue. So previously macrophage is sitting here, so as this is sitting here, so it is engulfing the bacteria and degrading it, and it releases some components which are most important in this case, some interleukins like interleukin 1, interleukin 6, and TNF alpha or tumor necrosis factor alpha. Now these three things are the most important chemical mediators during this inflammatory response. Okay, so they are releasing these components. So this component will act on to the endothelial wall and tell this wall to increase the permeability for other cells, right? Now again, other cells like mast cells, they chew up, and after gaining the signal from from these macrophages and other cells, they start releasing some components like histamine. I've told you before, histamine, prostaglandin, and leukotrienes. Now this histamine, prostaglandin, and leukotrienes, they are going to work onto this cell, this endothelial membrane again. And what they are doing here, they are again telling them to increase the vascular permeability, and also they start to damage some of these vascular endothelial tissues. Okay, and the third, so this is the second response, and the third response that we can see is that from the tissue damage that we have finally looked in the previous slide. This is due to the tissue damage. We have seen the production of bradykinin, fibrin, and also plasmin. Now all of these components will come here. You can see bradykinin, fibrin, and plasmin, they will come and they again act on to this uh, endothelial wall, so that the endothelial wall permeability is getting increased. So increase in permeability, this is important, right? Permeability. So as a result of increase in permeability, cells like say white blood cells, which are there to fight against infection, like neutrophils, like lymphocytes, like T lymphocytes, B lymphocytes, they can migrate in. Now for their migration of T lymphocyte, B lymphocyte, and neutrophils, they must need two important type of interactions. So let me tell you, two interactions are important. Now The first interaction, the important, is using selectins. Selectins, this is the first important thing. Now what are selectins? Selectins are some chemical components found onto the surface of all of these cells like neutrophils and lymphocytes. Now the example of selectin is P selectin found on platelets, V selectin, and different type of P selectin, V selectin, and T selectin. All these type of selectins are found onto the surface of this cell, and also these selectin receptors and are also found onto the surface of this vascular endothelial wall. So there is an interaction between this selectin and vascular endothelial wall. Now this interaction between the selectins, between the selecting, actually telling us all the cells to slow down. So it is providing the signal to slow down a bit. This is the first type of signal that they are getting. Now the second type of signal that they are getting, they must stop, right? Slowing down is not important, not enough. It need to stop before entering into this tissue, right? So they need to totally immotile, right? And in those conditions they require another mode of signaling using another type of, let me change the color, another type of chemical mediator, and that thing is called integrin. So let me write, it is called integrin, right? So this is called integrins. Now these integrins, the example of integrin is, you can see here, VLA and LFA. Okay, so VLA, this should be VLA, very late antigen, not VL. And obviously LFA, leukocyte functional antigen. So these are the important chemical mediators or structures, receptors that are coming out from monocytes and lymphocytes and all these cells. And PSGL is another type of integrin. Now this integrin, with using this VLA4, LFA1, they can interact with other type of integrin receptors that are found on the endothelial wall, which are VCAM1 and ICAM1. So we can see the integrin interaction of LFA1 with ICAM1 and VLA4. It should be VLA, not VL, VLA4. Okay. So VLA4 with VCAM1, and this interaction is important. After this interaction, it actually pause and stop those cells, and as those cells are stopped at this place, then they can move through this junction, or you can see there are a lot of cell lining are there, so cells are having gap between them. Now due to the release of this kind of factors from macrophage, histamine, as well as from the tissue breakdown products, they increase this vascular permeability. So as you can see, there is an increase in vascular permeability, and now the activity of selectin and integrin is actually making all the cells stop. Then they can enter through these junctions, small gap junctions that are formed between the cells. Via this process is called diapedesis. Now due to this diapedesis they can move. Now the movement is not done because there are also chemicals like this C3A, which is a complement factor, C5A complement factors. These are called anaphylotoxin of complements. These things and also other things like histamine and all these things, they can act as chemotactic factors. So let me write it here, chemotactic factor. Now this chemotactic factor are helping all these cells to migrate toward their direction, right, via the process called chemotaxis. Now via the process of chemotaxis they will migrate towards those release of those particular chemicals, so that they can reach finally to the bacterial cell and they can start doing the further work. And what are the further work? You need to study all the rest of the immunology for that, right? So this is how the whole process initiated. And when this process is going on, you can imagine the release of all these materials call upon of all the cells, it will lead to swelling of the place. So this is a symptom. And obviously due to the result of red blood cells moving, and obviously this vascular permeability, some red blood cells can also migrate through this place. So we can see red blood cells coming onto the tissues. So it's also the result is redness, and obviously sometimes as a result of that blood can remove outside, so it's obviously causing the redness. And obviously loss of functions will occur because due to the result of secretion of some chemical mediators like leukotrienes, like leukotriene B4, C4, histamine, they can damage the tissue, so tissue damage, so loss of function, it also takes place. And obviously pain must be there because all these things are going on, and pain and heat will generate due to this process, right? So we can find that why we can see these 5 different signs are associated with this particular inflammatory response, right? Now let us talk a little bit about the systemic inflammatory response. We have already talked about inflammatory response in the acute phase, but in systemic phase, first the acute phase response set place, and once the acute local inflammatory response set place, it can provide signal to our brain, it can provide signal to our liver as well as to our bone marrow. So it can trigger the signaling. Now as it is triggering some signaling to our brain, liver, bone marrow and kidney, from those different major organs we are having more different level of expression, because we require this kind of systemic inflammatory response if a same kind of infection is staying there for a longer period of time. But remember, this is not equal to chronic, so this we can't call it a chronic. Chronic means in those cases all the time it is going on, but this is not chronic, but still this can be systemic. That means it can move the signaling from one place to another place. Now for example, this local acute inflammatory response provide the chemical mediators to the brain, and the mediators here I've told you before, in all this kind of inflammatory response, three important components: interleukin 1, interleukin 6 and TNF alpha. So using them they provide the signal to brain and obviously the hypothalamus of the brain, and it starts secreting prostaglandins, and as a result of high secretion of prostaglandin we end up with high fever. Now remember, I've talked before that systemic inflammatory response can be related to fever, right? Now then, this brain can also send some signal from the pituitary gland of the brain. It can start increasing the concentration of adrenocorticotropic hormone, or ACTH, which will go and induce adrenal cortex to finally release some corticosteroids. Now this corticosteroid can lead some effect on the liver. So as the corticosteroid come and signal the liver, the liver will start to produce acute phase proteins like C-reactive proteins or CRP, and obviously serum amyloid protein or SA. So these proteins like CRP or C-reactive protein, serum albumin, what you can say, amyloid A or SA. So these are the proteins that are being produced by liver cells due to the response of this corticosteroid hormones, right? So this is a kind of response. Now the second kind of response, so this is first. The second kind of response we can see is the direct response from this local inflammatory site to liver, and this response again via IL-1, IL-6, TNF alpha, right? And it again triggers liver to produce the CRP and SAA proteins. Now the third kind of response is can be towards the bone marrow, again using the same chemical mediators like IL-6 and TNF alpha. It is telling the bone marrow to increase the CSF by stromal cell and macrophages, and as a result of that it start increasing the secretion of white blood cells. So white blood cell concentration start to rise. So that's why I've told you that during the chronic inflammations, because systemic can be modified to chronic, we all know systemic can be modified, sorry, to chronic infection. So in those conditions we can see at the very beginning they can signal to this bone marrow that start producing more and more white blood cells. They can signal our brain to produce prostaglandins so that it will turn out into fever, and obviously it can start secreting all those corticosteroids and instigate the liver to make this kind of proteins like CRP and CAA or SA, sorry. So these are the process. Now this is at the end. So in case of chronic inflammation, we have seen in case of both the case of acute inflammation, obviously systemic inflammation, the first response begins with 5 typical signs, but after the 5 signs it also modified into fever. We have seen that it modifies to fever, and obviously not only fever but also it modifies to say what we have seen, increased level of WBC. So these are again important uh, what you can say indication of chronic inflammation. But also except for that there is another important part, which is formation of large cell or large activated macrophage cell. Now what we know that if there is an infection, so there is a bacterial cell, macrophage are the larger cells, the macrophagocytic cells, so they will start engulfing them. So this is the macrophage, they start engulfing this bacteria. So once after engulfment of the bacteria, they can become activated, they can release some of the important chemical mediators like IL-12 and all these things to instigate T helper cells, and then T helper cells in turn can start to release interferon or IFN gamma and all these things. So this is how our whole system actually works. But once it is becoming activated, all those macrophages can stay there for longer period of time. And if there are a lot of concentration of a particular pathogen like a bacteria, for example in case of a TB infection or tuberculosis disease, in those cases a lot of tuberculosis bacteria are present there in our alveolar regions of our what you can say lungs. And in those cases, all of these activated macrophages will start to fuse with each other. They start to come closer and start to fuse with each other. And as a result of the fusing, what they produce is a large cell, which is a polynucleated cell. So you can see here one large cell is being produced here, which is having more than one nucleus, and many bacteria are trapped inside. And obviously these activated macrophages are also destined to produce, and they are destined to fuse together to make a even larger activated macrophage cell. This is called polynucleated polynucleated macrophage. So this polynucleated macrophage can be found in case of chronic inflammations, right? Now for the disease like TB, that can be present for throughout your lifetime. So in those cases, this kind of thing can occur, right? So that's it, and I hope that's helpful. Thank you. Sorry, I forgot to mention the biochemical response of pathogen infection. I must tell this because this is also an important part, that whenever we've seen that the different level of those inflammatory responses like acute inflammation or systemic and chronic inflammations, we must have some biochemical responses of this pathogenic infection. Now, what are the biochemical response that we can get? Now, in this case, what we can see, membrane phospholipids are playing the most vital role, and those after the activation of phospholipase, it will cleave some membrane phospholipid, and what it can generate two important components. One is arachidonic acid, another one is the lysopaf. Now this paf is a platelet aggregation factor, right? Now whenever this lysopaf is produced, from this lysopaf, PAF or platelet aggregation factor can be generated. Now as the result of platelet aggregation factor generation, it will call upon platelets as it call upon platelets. So all the platelets will come, they'll start lining together using fibrin and all these things. They can form what we call a blood clot. So this is the biochemical process of blood clotting, which is helped by, or it is associated by this PAF. And the second mediator, arachidonic acid, can even further divided into or further produce two important components via two different pathways. One pathway is the cyclooxygenase pathway. Through this pathway, it can generate prostaglandin G2, and through the lipoxygenase pathway, it can generate what we call leukotriene or leukotriene A4. Now this is from arachidonic acid. Now this prostaglandin G2 can even produce prostaglandins and thromboxane, and obviously leukotriene A4 can further produce different varieties of leukotrienes like leukotriene B4 and leukotriene C4, D4 and E4. So these are the components. Now among all of them, these prostaglandins are helping in the vascular permeability and vascular dilatation, and obviously in neutrophil chemotaxis they're playing vital role. Thromboxane is also playing the vaso platelet aggregation of or other effects. Now on the other hand, this leukotriene family is much more dangerous. Now these leukotrienes, they can lead to the smooth muscle contraction of the bronchial muscle contraction and neutrophil chemotaxis. Now during this inflammation activity, there are several rounds of inflammation activity. Usually these prostaglandin mediators are formed and released into the first phases of those inflammation activity, and on the later phase, or this is first or early phase, so let me write it here, early phase, they are secreted in during the early phase, but these leukotrienes are secreted during the late phase of inflammation. Okay, so these are the distinguishing, and leukotrienes are much more powerful than those that are secreted by mast cells like histamines and all these granules, right? So these are the process of chemical response during a pathogen infection, and I hope now it helped you to connect all these concepts. Thank you.